In the first lecture, just for refreshing, we discussed about the basics of bioinformatics with few examples and the different features of bioinformatics, for example, development of databases, algorithms and hypotheses, structure based drug design and next generation sequencing. And we discussed about the applications of bioinformatics on different complexities of biological systems. If we talk about the DNA, it is genetic material and the information stored in the DNA allows organisms that are able to regulate their internal chemical composition, growth as well as reproduction. For example, it allows mother's curly hair and father's eyes and so on, that we know that getting the genetic information. So, the various units that govern these characteristics at the genetic level are called as genes. So, there are several genes and collectively form a genome sequence. It contains various genes. So, if you look into the genes, they contain specific sequence of nucleotides, for example, A, T, C, G, right, adenine, guanine, cytosine, and we take a nucleotide. So, this is an example. So, it contains a phosphate group here, and here this is the central deoxyribose sugar, and here is a base. Here there are 4 different bases for the DNA and the 4 different bases for RNA. 3 are the common and 1 is different, thymine replaced by uracil. So, if you look into this DNA as well as the RNA, so the name of DNA that stands for deoxyribose nucleic acid come from the sugar molecule contained in the backbone. So, if you see, this is the base, this is the phosphate, and here this is sugar. So, here if you see, this is starting from 1 here, 2, 3, 4 and 5. So, in the 2 prime here it is H, this is deoxy. So, hence the name deoxyribose nucleic acid, this DNA. For the case of RNA, so this is OH, so because this is oxyribose, so it is the ribonucleic acid. So, there are 4 different bases. In the case of DNA, we have adenine, cytosine, guanine and thymine. Right. If you look into these 4 bases, they are classified in 2 groups. One is called purines. Right. It has double ring shape. Okay, here is one ring, this is another ring shape with a pentagon attached to one side of the hexagon. So, this is the hexagon and this is another pentagon attached to this hexagon. So, in pyrimidines, they have hexagon shape here, right. So, made generally with the 4 carbons and 2 nitrogen. If you see 1, 2, 3, 4 carbons and 2 nitrogens. So, cytosine is both the DNA and RNA, and the difference between the thymine and uracil, okay, thymine you can see in the DNA and the uracil you can see in RNA. So, here this is the one CH3 group, there is the major difference between the DNA and RNA. So, difference comes from 2 different aspects, right. What are 2 different aspects between DNA and RNA? Right, one is at the sugar level and the second is on the base level. So, you can see the thymine and for the DNA and the uracil in the case of the RNA. So, how they form? So, there are the 4 different bases attached to the ribose sugar and the phosphate, and how they form the chain of this DNA sequence. So, here this, for example, take molecule 1, this is a molecule 2. So, in this 3 prime, so here this is OH, right. So, here this is the phosphate attached to this OH, right. So, this H and this OH have the condensation reaction, right. So, eliminate water molecule, and then you can see there is a linkage, phosphodiester linkage, because phosphate has 2 esters, right, one with this one with this one. So, you can form the diester linkage, so you get this molecule. So, if this is the first one, right, and here this is the second one, here this is the linkage with the condensation of this water molecule, right. So, when you have this phosphodiester linkage, here I show the continuous ones. So, the one nucleotide contains from here this line to this line. So, you can see this is the phosphate attached here, and this is the ribose, and here this is the base, right, and here up to this one you can see the one nucleotide. So, now if you look at this DNA molecules within the cells typically of 2 double strand. So, this is the one strand and this is the another strand, right. If the information contained in the one strand here, right, essentially redundant on the information on the other. If you see, this is redundancy is not the same, because the direction is, if you see here, the direction is from 5 prime 3 prime, right. So, here this is one is 5 prime 3 prime and the other one is under 3 prime 5 prime, and then you can see the complementary of these bases. So, every G on one strand, for example, right, and C is found on the complementary strand and vice versa. So, here I show the example, right. So, here is the green is adenine and this red one is cytosine and the blue is a guanine and the magenta is a thymine, right. So, adenine is paired with the thymine, right. Here this is cytosine is paired with the guanine and here this thymine is paired with adenine and the guanine is paired with cytosine. So, when they make this model of this DNA, first they try to put the two strands here, this is the backbone, right, phosphate backbone here, and then first they try to put the bases on the two other sides. Then they could not get the compatible structure. So, then they think about the ladder type structures. When the kids they play Legos, right, they like to link these Legos each other, right. So, likewise take the ladder type structure and they put the two backbones here and they make the side chains the bases in between the ladders, then they found it exactly matched. They could see the matching bases, right, with respect to the chemical groups as well as the space, right, and then they found the hydrogen bonding pattern also they could exactly fit with the favorable energy. So, now for this one is the Watson Crick the proposed model for the double helix of DNA and they got the Nobel Prize in 1962 for this double helical structure. So, now how this base pairing happens? So, if you have adenine and this thymine, right, it forms two hydrogen bonds, right, this pairing, and the guanine and cytosine they are paired with 3 hydrogen bonds. Why this pairing is specific? Why not with the other bases? Why A is not pairing with G and the T is pairing with the C?
Because of this space time state like 11 angstrom distance some because one is purine and another one is pyrimidine, so that will be like uniform space.
Right. So, because if you right. So, if one is purine and one is the pyrimidine, if you have try to have this type of two purines or two pyrimidines, either they have extra space which also has loss of energy and also they are very crowded. So, they due to steric interaction they are not able to pair with each other. So, two different ways, one is the steric hindrance and the second one is the chemical group. So, because of the two reasons adenine is always pairing with thymine and the guanine is always pairing with cytosine. So, if you know one strand then we can know the complementary strand because A always pair with the T and the G always pair with C. So, if you see this one as I discussed now, the two strands of DNA molecule are complementary, right, but they are not in the same 5 prime 3 prime direction. One is in the 5 prime 3 prime direction, one is in the 3 prime 5 prime direction, right, because most of the cellular process they involving the DNA occur in the 5 prime 3 prime, this is why they write the sequences in the 5 prime 3 prime directions, right. Okay. So, now if you have one sequence, for example, ACG TT ACG, okay, this is a sequence. What is the complementary sequences? If this is a 5 prime 3 prime sequence, right, when you go to the just complementary, then we can get the pairing, we can write the complementary sequence, but that will be in the 3 prime 5 prime direction, right. So, for the A, what is the complementary for A? T, for the C, it is G, right. So, we write like this A to T, C to G and G to C, T to A and so on. This is the 5 prime 3 prime direction and here this is 3 prime 5 prime direction. So, we need to the complementarity, so we have to reverse the direction. So, if you write the 5 prime 3 prime then we reverse this direction, right. So, then start from C G T A A C G T, right. This is how to write the complementary sequence. Here I show the example, this is A, this is C and here this is T and here this is G, right. So, this is here we write 5 prime 3 prime, this is 3 prime 5 prime, this is why just you can see the complementary here. So, now I give this example, okay, this is a sequence. What is the complementary strand from 5 prime 3 prime direction? G, C, T, A, G, C, A, G, A, T, T, A, G, C, T, T, A, C, G, G, C, T, right. So, we have to get the pair and the reverse direction. So, right, this is the complementary strand. Okay, now in bioinformatics you can write algorithms for any given sequence, you can write the algorithm to get the complementary strand, right. How to write the algorithm? First what you have to do?
You have to take the sequence as input.
Take the sequence as input, reverse it and the complementary, or you can get the complementary and reverse it, right. So, you can do that. Likewise, there are various programs available in the literature, right, because they collected various programs to analyze the DNA sequences, right, and put it together as kind of a package. So, EMBOSS is such a package. EMBOSS stands for European Molecular Biology Open Software Suite, right. It is a compilation of several programs. You can see the several programs at the end, at the side here, right, the menu. So, for the sequence analysis, so if you have a DNA sequence, you can use the sequence and carry out various analysis, right, using this specific software called EMBOSS, right. How to get this complementary strand from EMBOSS software? So, you go to the EMBOSS website. So, then go to the edit and finally, you can see the module RaveSeq, thus reverse the sequence, reverse and complementary a nucleic acid sequence. So, either you can give a sequence from database, or here you can choose a file if you have your file in your computer, or you can give a sequence, like manually type a sequence. Here I give a sequence ACT GACC, right. So, now if you click on the run reverse sequence, then you will get the sequence GGT CAGT, is it correct? Right, because we just inverse complementary of this sequence, right, that is fine. Now, you get the complementary strand. Now, next step is when you have the DNA sequence, you can translate this into proteins, right. What are two steps involved in protein synthesis? One is the transcription and second is the translation, right. In the transcription, what happens in transcription? DNA is converted to messenger RNA by transcription by messenger RNA, right, RNA, and the translation messenger RNA to the proteins, right. So, two steps. First one, we have to have the DNA and a change to RNA, right. In this case, DNA has 4 different bases A, T, C, G. In the case of RNA, A, U, C, G, right. So, this way if you see here, this is the DNA sequence, it contains A, T, C, G, right, but this is RNA sequence. This has no T, but instead of T you have U, right. The complement of A is T, instead of T you put U here. C is G and this is G and this A is U and A is U and so on. Now, if you have the RNA sequence, right, now then this RNA is translated into proteins by ribosomes. For DNA transcription, mainly the RNA polymerase does this transcription, right. Now, the ribosomes are responsible to convert this, translate the mRNA to proteins, right. So, there are the different nucleotides, right. They use the codons, there are 3 nucleotides together form one codon. So, each codon they code for a specific amino acid, right. So, there are 4 different nucleotides, right, but how many amino acid residues? 20, right. So, 1 to 1 correspondence is not possible. If you take 1 to 1, so 4 nucleotides will code for only the 4 amino acids, but we have 20. If it is combination of 2, how many combinations? 4 into 4 equal to 16. So, that is also not possible because we have 20. So, these have the 3 combinations. 3 means totally how many possibilities? 64 possibilities, right. So, 3 into 3 into 3, right. So, if you give the 4 into 4 into 4, 64 combinations, but 20 different amino acids. That means, there are several codons which code for same amino acid, that is called a degeneracy, right. Not one single codon to one, right, same different codons they code for same amino acids. If you see the phenylalanine, right. So, there are 2 codons UUU and UUC, they code for phenylalanine. For leucine you have 6 here, UUA, UUG and CUU, right, CUC, CUA and CUG, okay, all these code for leucine. So, as a one stop codon, what is a stop codon? You can see here this is a stop codon, right, UG is a stop codon. Also tryptophan is coded with the only one codon, right, UGG. So, this is also one of the reasons why tryptophan is occurring very less in the case of the protein sequences. So, 18 of the 20 amino acids are coded with more than one codon, right, and this is called the degenerative system. So, now if we have a DNA sequence, can we get the protein sequence if we have a DNA sequence or RNA sequence? Here I gave RNA sequence. Can we get the protein sequence from this RNA sequence? Yes. What to do? Yeah, first take 3, right, take these 3, right, these 3 steps, right, fine. So, ACG, what is ACG? A here, C here and G here, right, ACG. So, AGC is for the 3 right. So, the ACG code for T, then UGC, that is cysteine, right, UGC, UGC, right, UGC is here, this is cysteine. So, you can put cysteine, right. Then GCA is for the alanine, and this again UGC for cysteine, and AAC is for the asparagine, CJ is for the arginine, and AUG right, AUG is for the methionine, right. So, now there is end, so they put the X, right. So, in this sequence what will happen if you delete this nucleotide A, if this is not there, you will get a similar sequence or different sequence? Different sequence, right, because now the codons are CGU, GCG, CAU, GCA, ACC, GAA, UGA, right. So, now CGU code for CGU arginine, right, this arginine, this is alanine, this is histidine, GCA alanine, T, GAA, GAA, E and UGA, UG, the stop codon, right, they put a stop codon here. So, if you remove one single nucleotide, so this is totally different, right. So, if you this way, if you have the DNA sequence and we look for the protein sequence, we should know exactly where they start the codon, right, otherwise you will get the different protein sequence. So, there are various resources, you can also use programming, you can write your own code, right, to get the translate the RNA sequence to protein sequence. How to write a program, right. So, first what information required? Input sequence, you need this input sequence, right, you need the, we know the RNA sequence here. So, you need to get the protein, right. So, how to write the program to get the protein sequence from RNA sequence? What are the input necessary? First is RNA sequence, right. One is first we need the RNA sequence. For example, ACU, GC, AU, G. Okay. So, you have the RNA sequence. Then what are the other information you require? The code, right. So, you get the mapping, right. You have the table, right, the codon to amino acid table, right, codon to amino acid table. Right. So, then you will get the information regarding each codon codes for which amino acid. Then what you have to do? Then first change the RNA into triplets, right. You have the codons, right. So, 3 nucleotides. So, you cut into 3 pieces, right, not the overlapping, right. So, here you have to stop and then start from the next one. So, non overlapping, right, that is very important. Then we have to match, right, with the codons with the table, right. Then you have to match the sequence and then finally obtain the amino acid, right. Then finally, if you do it till the end of the sequence, then finally you get the protein sequence. So, you get the RNA sequence, read the RNA sequence and get the table, right, the codon to amino acid conversion table, and you have to cut the RNA sequence into the pieces of 3 nucleotides, right, for each codon, right, as mainly non overlapping ones, right. Then you map with the codons to the amino acid in the table, then you get the amino acid, right. Then finally, you do till the end and you get the protein sequence. So, now several resources are available in the literature. So, again you can use the EMBOSS software to translate the RNA sequence to protein sequence, right. So, here you can use the transec, this is the module available in the EMBOSS software, right. So, here you put the sequence ACG, UGC, GCA, UGC, AAC, CGA, UGA, right. So, now if you go with one, so it will ask for the different frames, right. So, there are 6 different frames, right. How many frames? 6 frames, right. So, what are 6 frames? 3 forward frames and 3 reverse. I will just explain the next slide, right. So, at the moment you take this sequence and if you asked to convert the protein sequence, right, you can see this into this same sequence TJ. Okay, I showed the example here, the same sequence I give, right, ACG, right. So, I get the same sequence TCACNRMX. So, if you cut this one, write the first one, then you get this same sequence. So, for each sequence we have 6 reading frames, right. How to get the 6 reading frames? 3 in the forward direction and 3 in the reverse direction. How to get the 3 in the forward direction? For example, this is the 5 prime 3 prime, this is a DNA sequence, right. So, first you cut into codons, 3 nucleotides, okay, they cut it into CAA, TGG, CTA and so on, okay, the DNA. So, now we go with these amino acids, then CA codes for Q and TGG code for W and this code for L. So, you will get the amino acid sequence, right, this is one. And the second one, you take this one out C, then you make into another 3. So, AAT, GC, TAG and so on. So, now you check this table for the amino acid. So, here you can see the amino acids. And the third one, if you take these 2 C and A, right, and then you start with the CA, ATG, GCT, AGG, TAC and so on. So, now look into the table of each codon, right, you will get the M, A, R, T and so on. What will happen if you move one more again?
Again
the same will repeat, right. So, if this is put it as TGG, so it will start from here, right, you do not have this amino acid, but you start from the same sequence, right. So, in this case we will get 3 different reading frames. Likewise, in the reverse frame. So, how to get the reverse frame? Reverse complement, right. So, if you have this 5 prime 3 prime, now if you take the complementary, right, then if you take the complementary, then you will get the 3 prime 5 prime direction, right, and you reverse it, right, then you get the same strand from the 5 prime 3 prime. Then from this 5 3 prime sequence, then you make the codons, right, here CTC, GGA, TTT and so on, right. This is the, this will code for L, GGA code for G and TTT code for F, right. So, now you can get the amino acids. Likewise, just you take one out, then you will get the second frame and the third frame. So, the total you will have 6 reading frames, 3 forward frames and 3 reverse frames. So, you can use this transect, right, to get the 6 reading frames. So, here if you give the input sequence and then here you have the options, whether this is the forward reading frames or the reverse reading frames, right, or everything. So, if you go with the all 6 frames, right, you get here a click on all 6 frames. So, you will get the sequence, right. So, this is the forward frame 1, this is 2, this is 3 and the reverse 1, reverse 2 and reverse 3. So, then if you go into this same software EMBOSS, so if you see there are various tools available in the EMBOSS, right. So, everyone have the different different values and they have used different algorithms to get the values. So, you can use any of these different module available in the EMBOSS, right, to get the characteristic features of this protein DNA sequences or the RNA sequences.